Welcome to an all-new lecture for the Clinical Biomarkers module. Today we're going to be talking about inborn and acquired mutation as biomarkers of disease. I think this is going to be a really interesting tour. We're going to cover a lot of ground though, so hang in there. We put in an intermission about the halfway point in the lecture. We're going to start with some overview of some terms from genetics that will help us to understand this topic a little better. So we're going to start there, and from there we're going to talk first about some of the genetic variation that we are born with that may determine our predisposition to particular diseases. The emphasis in today's lecture is definitely going to be on predisposition, not deterministic disease where your genes say you shall be sick at this particular age with this particular condition. So predisposition is a very strong class that this is going to apply to. So we're going to talk about this in the context of human disease, talking specifically about newborn screening, and also about the reasons that take people to do genetic testing as adults as well, opt-in screening. From there, we're going to have, after our intermission, a discussion of acquired mutations and their relationship with disease. We're going to start with some overview on a concept of driver mutations in the context of colon tumors. From there, we'll talk a little bit about the genome lots of people never think about, their mitochondrial DNA. And finally, we'll move from there to discuss infectious disease and its relationship with its own acquired mutations. I want to start by correcting a common error that we make in discussing genetics in popular language. You'll sometimes hear people say, oh, I've got the breast cancer gene, and so on. And that kind of language actually makes a pretty serious mistake. All of us have nuclear genomes that have essentially the same set of genes overall. What differs among us, however, is which alleles an individual has. So I may have, I just like most other humans, have a BRCA1 locus, the gene first associated with breast cancer. But the allele I have for it may be wild type, or it may be one that predisposes me to disease. So we shouldn't say that we have the gene. We should say that we have an allele that predisposes us to a particular gene, a particular disease, if that's where we get. So an allele then is a particular variant that we find for a particular gene sequence. Now, because we are diploid organisms, we have two copies of every chromosome. Therefore, I might be homozygous, meaning that both copies of the gene I have are wild type, or maybe both disease, or I might be heterozygous, where the genetic alleles that I have are different at this particular location on my two chromosomes. So homozygous or heterozygous may have very different phenotypes attached to them, or they may have very similar ones. If we think about a case like cystic fibrosis, I might have two wild-type alleles, or I might have a wild-type and a CFTR, a cystic fibrosis-associated allele. Both of those are healthy phenotypes. But when I'm homozygous for the cystic fibrosis variant of the sequence, then I have the disease. That's a deterministic case. So homozygosity, having the same alleles, or heterozygosity, is something we should all understand. Next, I want to talk a little bit about haplotype. If you have a father who is heterozygous, having the orange allele and the brown allele for these two chromosomes, and a mother who is heterozygous, who has a green and a magenta allele for a particular for these chromosomes, you can imagine that each of their children may get one or the other of the father's chromosomes and one or the other of the mother's chromosomes to then be some combination of those colors. So in this case we see that there are 4 different combinations where the mother contributes magenta and the father contributes blue, the mother contributes green and the father contributes blue, etc. So we see then that the heterogeneity in the parent's chromosome lead to this combination process that we see in the children's genetics. Now there's another aspect, that the haplotype aspect reflects the fact that the father is not at each gene giving a different a different chromosomes contribution. We don't see, for example, that child 1 has 50% blue and 50% orange in the chromosome received from the father. And that's a slight misconstruction that we see in this diagram. This does not reflect recombination, which means that we may see a mixture, we may see crossovers between the father's 2 chromosomes to reflect the composite chromosome that goes to the kid. But those crossovers generally are not going to be tens of thousands of crossovers between this pair, so that we end up with very much a mishmash of orange and blue alleles contributed by the father. Generally, you're going to have far fewer numbers of crossover events that take place between a pair of chromosomes. So this leads to a common receipt of multiple genes that are of the alleles that are all close together. So if you have a crossover that takes place just once at the midpoint of this chromosomal pair, we see that the child would receive orange for all of the top half of the chromosome and blue for all of the bottom half of the chromosome. And that's very reasonable. But 1 or 2 crossover events between a pair of chromosomes is not going to lead to a subdivision of a partitioning of that chromosome into thousands of different segments where it switches randomly back and forth between blue and orange. This creates a linkage, in effect, an association among these genes that reflects that the further apart they are on a chromosome, the more likely they are to be separated by a recombination event. This means that because the father has these particular alleles in this orange region, they are more likely to be inherited together. If they are close together in the blue chromosome, they're more likely to be inherited together. So this variation coming to us, where genes that are closest together have the same alleles coming as a package, we call that a haplotype. And often these haplotypes are very descriptive in what kind of inheritance takes place. Now, as I said, the greater part of today's lecture is not about disease that necessarily comes about because you've received 2 alleles for it. Instead, we're going to be talking about genes that predispose us towards having a particular phenotype. So this term penetrance is one of the first that comes to mind here. We see that people who have a particular genotype tend to be more likely to be sick. Okay, so there's some percentage of individuals who have a particular genotype that exhibit that particular illness, or in this case, the phenotype. But it's not deterministic in that not everybody who has that genotype becomes sick, or not everybody who has the wild type stays healthy. So this penetrance then is a percentage that we use to talk about the extent to which genomics are pushing their way through to create an outgoing phenotype. What we are seeing instead is a predisposition or susceptibility, which is to say there's an increased likelihood that this person will develop a disease based on the alleles that they have, the genotype that they represent. So you might ask yourself, well, if genes aren't, if the alleles a person possesses are not the thing making them sick, what is? And I really felt like this diagram from a paper on autism spectrum disorder was a really useful reminder of just how complex the biological interactions are that lead to our health or our disease. So you can see here that you may have mutations and genetic susceptibility that lead to an increased likelihood that you have disease. There are a lot of other things going on. Epigenetics, for example, has become a very fruitful place to study, where we notice that the methylation or acetylation or other kinds of modifications on histones can make a big difference in gene expression. The direct methylation of DNA can have a very big impact on gene expression. So epigenetics has a lot to say about how those alleles will play out. Copy number variation reflects the fact that for some people you may have multiple copies of an allele, or you may have expansions based on repeats that appear within a gene. We'll talk about an example of that in some of the slides ahead. So copy number variation could lead to an increase in expression just because there are more copies of the DNA there. Environment is obviously a big deal. Environment is more than did Dave hike a mile today, or did he have orange juice for breakfast. Environment can take into account things like smoking. They can take into account what medications I take. Am I on beta blockers, for example, for blood pressure? They can have a lot to do with what kind of microbiome I have. If I have a preponderance of a particular bacterium growing in my gut, I might be more or less resistant to a disease that could be associated with it. So environment can be wildly complex. And there's also plenty of sex-linked modifications. If I'm a male or I'm a female, I'm going to have a different kind of response to alleles that are found on the X chromosome. That's certainly an issue. But just the way that the hormones my body started producing when I was growing up are going to impact how genes get expressed. A woman who has almost exactly the same genotype as I do might respond differently because of the hormonal environment of growth. So all of these factors can come into play in determining whether genes are going to result in, are allowed to result in disease or not. And we can't simply say that, oh, I've got the gene, the allele, for a particular disease, and that just automatically means I'm going to get sick. It's not quite how it works in real life. I think a lot of people are familiar with the term GWAS, a genome-wide association study. A GWAS is designed for us to understand which genomic variants that may be observed for a given person, how those, which of those variants is most strongly linked to the occurrence of disease. And you might think, well, gosh, we've had GWAS now for a couple decades, so surely we know lots and lots of bits of DNA that are associated with the occurrence of disease. And we do. If you go to the GWAS catalog, for example, you will see hundreds of studies, maybe thousands, frankly, that have now been conducted to link genetic variants to disease. See, I just altered my environment there by having a sip of lovely masala tea. Thank you, dear. So, what we see then is that GWAS has had successes, but those successes are somewhat limited. And I wanted to point you to odds ratios as an example of the kind of output that you can get from a successful GWAS. In an odds ratio, we're drawing an association between genotype and gaining disease in this case, using a pair of cohorts, people who are sick, people who aren't sick. And I'm going to take you through a little bit of math, and I hope you hang with me because this is an important thing to understand. These odds ratios compare the proportion of sickness among people with disease genotypes, or with putative disease genotypes, and wild-type genotypes. So you can imagine then that we have cases, these are people who are sick, who have the genotype we think that we're hypothesizing is associated with that disease. We have people who aren't sick, but who do have this genotype. So this is the example of somebody who has a breast cancer allele for BRCA1 who doesn't then go on to have breast cancer. That's a control in this case. So we can see then that this fraction is to say, of the people who have this allele, what fraction of them are sick? That's this A versus A divided by B part. Down at the bottom, we have the same fraction. We're asking what fraction of people are sick, this time counting just the people who have wild-type alleles. So that's X versus Y. So you can see then that if the numerator, the top part of the fraction, is a lot higher than the bottom part, the odds ratio is high, suggesting that of the people who get this, the people who have this disease allele have a much greater chance of developing illness than the people who don't have it, who have wild type. Okay, so the ratio that falls out, if the odds ratio is very near 1, there's very little association that we see. If the odds ratio is very high, we would like to be able to convert from an odds ratio into a p-value. So frequently, we basically say that if the odds ratio is very far away from 1, we can use the chi-squared test to turn that into a p-value to say the probability that by random chance this value would have drifted this far away from 1. And that can be compared to kind of a genome-wide threshold, taking into account all the different, just the size of our genome and how many different recombination units there are within it, to say that if that p-value falls below this threshold, we're going to call it a strong association, that this gene has a real impact on whether or not someone develops disease. I'm sorry, I've placed that in far too causative language. To say that a genetic variant is associated with the disease is not to say that it causes that disease. It could just be a landmark that's pretty close by, a bit of DNA that does have some association, have real association with the condition. But I should be much more careful in saying that having that allele does not necessarily cause disease, it's associated with disease. I hope that distinction makes a difference. Okay, so how does, if GWAS is such a wild success, what are the things that cause people to say, but what did we really accomplish there? I want to take you through 3 concepts that kind of make this space blow up a little bit. The first of these is the missing heritability problem. When we do GWAS, we find large sets of markers that are associated now with many different phenotypes, but we see that a large number of people get this particular illness that don't have the genetic markers we associated with it. That's a case of missing heritability. You can read a lot more about that in this paper from PLOS Genetics. There are lots of cases where disease takes place, but we don't have genetic markers to point the finger at and say, this person is sick because they have this allele, and this allele. Those SNPs that were present, those are all markers for this disease, therefore we know why this person got sick. There are plenty of cases in which disease occurs and we can't explain from a genetic point of view what's contributing at all, or what's associated at all. So missing heritability is the first of these. Next, familial aggregation. We have this problem that many times we will find that diseases, rare diseases, are not just found higgledy-piggledy across the population, but rather certain families are much more likely to carry these diseases. The trouble is that when you're trying to determine which markers within a person's genome are associated with disease, if all the cases are falling within a family, they have a lot of markers that are being passed down through generations that are close together in haplotypes. And trying to figure out which part of that inherited DNA is contributing, is associated with disease can be very, very difficult. Okay, so familial aggregation is one of the things that means that even though we may have lots of cases, because they're distributed narrowly in a few families, we lose resolution in our ability to pin down a particular bit of DNA associated with it. And finally, epistasis. Epistasis is kind of a confusing point, but I would just say that if you have millions of different loci across the genome, millions of different locations, you may have a situation where it's not just one genetic region that contributes to your chance of having this disease. You might have many different locations, and they're all interacting. That if you have two disease alleles, if you're homozygous for a disease allele at this location, but you have at least one wild type at another location, it's able to accommodate the miss that you have in this pair of disease alleles and compensate for it. So you may not get sick, even though you are apparently, at the one site we do know about, have two disease alleles. Because our genetic systems can compensate for damage in one place by the genes that are present at another place. This epistasis can lead to some very big complications in trying to pin down the particular effect a particular bit of DNA would have on disease condition. So, having spent a little time talking about genetics, let's talk about newborn screening. We need to be very, very responsive when a human is born to make sure that they've got the best shot at life that they can have. And lots of newborns out there are born with rare conditions that we need to know about right away in order to prevent any damage occurring from some of the genetic problems that humans are born with. So for that, we have newborn screening. I would note that one of the common ways that we screen for this happens before birth. So amniocentesis is a way that we're able to get a few cells from the developing fetus at 15 to 16 weeks. So this is about nearing the halfway point of gestation for a fetus. So you can also do things like chorionic villus sampling that can give us a bit of genetic information about the growing fetus at 11 weeks. So that's really brilliant, and these methods give us the information we would need to detect trisomy, for example. So in trisomy, instead of having 2 copies of a chromosome, of a particular chromosome, that growing fetus has 3 copies of the chromosome. And that can be really problematic, as we'll discuss in a moment. We can have aneuploidy or fragile X. There are all kinds of things that we can detect through doing a karyotype. We'll show you an example of what that looks like coming right up. There may be sequencing variants that we observe, things like the detection of sickle cell hemoglobin in the fetus. You might have the CFTR locus. We've already mentioned that as an example. CFTR, cystic fibrosis, can be a really serious disease. It used to just kill large numbers of people. Where we knew that they were sick, we knew why they were sick, and we weren't really able to do anything about it. But today, cystic fibrosis is no longer a death sentence. And this is a change that has come about basically in my lifetime. We detect it earlier, we have better methods for resolving the symptoms of the disease. We're getting a lot better at this. So being able to detect disease at a very, very early stage of development is giving us a better shot at seeing that those folks have a shot of normal life. And that, I think, is a brilliant success. Okay, muscular dystrophy is an example of one of the things we look for. It's a sex-linked trait here. Tay-Sachs disease, critical congenital heart disease. Obviously, if you're born and depending on your own heart for circulation, and your heart isn't functioning, you're going to have a rough time of it. So being able to detect these problems as early as possible is hugely important. Now, I mentioned karyotypes in the prior slide. This is an example of what that looks like. So here we're looking at an example of Down syndrome. I think it's a very commonly known trisomy, where 3 copies of a particular chromosome are present. We see that it's a trisomy at chromosome 21. So we've got the chromosome, the autosomes, the non-sex chromosomes are kind of sorted in order of size. So number 1 is the largest, number 22 is the smallest pair, and here we see that 21 is present in not 2 copies, because we're diploids, but rather in 3 copies, because this non-disjunction has taken place. So you might ask, well gosh, wouldn't it be better to ensure in the first place that our babies are never going to have 3 copies of a chromosome? But of course, most of us are expecting that everything's going to just go fine in the case of our pregnancies. So in a case like this, we find that some of the best predictors we have are not genetic at all. Mothers who are at higher ages have a disproportionate chance of producing a trisomy 21 in growing fetuses. So particularly if you're thinking that you want to start your career and get going with that, and babies we can hold off until you're in your 40s, like my age, then in a case like that, you'd better be thinking about the increased chance that is associated with trisomy 21 that's associated with that choice. So trisomy 21 is a very commonly used one. Today we see that we do screening via a few different proteins that can appear, and we also can use ultrasound imaging to detect developmental disorders associated with this, so that's also a plus. But we see that these measurements that could be made as early as 11 weeks in the pregnancy at the chorionic villus could allow us to do a karyotype that would detect this at a very early stage. All right. Of course, we should be thinking about things like sickle cell anemia as well. Sickle cell anemia is the result of having an abnormal hemoglobin. But to say that we just have a wild type hemoglobin and a sickle cell hemoglobin and that's it is much too simple a scenario, actually. when we do measurements to detect sickle cell anemia, we do not typically start by sequencing. I know we're in the biotechnology department. I know that sequencing everything is supposed to be the answer, but sequencing everything is not the answer. In this case, hemoglobin is a really common protein. If you take a little bit of blood from someone, you can rather quickly run a gel of the hemoglobins that are present in that sample to determine what the composition of this person's hemoglobin would be. So we start with hemoglobin A. This is the adult type that someone like me is going to have almost in exclusion of everything else. F is the fetal version. So if you were looking at a blood sample produced even before birth, you would expect to see a large amount of hemoglobin F rather than hemoglobin A to represent this difference. There are several genetic variations that have all been associated with Sickle phenotype. So we can use this electrophoresis to cause the S to migrate differently than do the A hemoglobin types. And there's also another type called C. There's actually a whole alphabet soup of them. But all of these four are kind of the most common types that we see. So a C-type hemoglobin can also cause anemia when the person has two alleles for this C-type hemoglobin. So when we see a nice prominent strand at the S hemoglobin, and we see that the person does not correspondingly have hemoglobin A to back it up, then we can suspect that sickle cell anemia is the cause here. Helpfully, we are now able to use metabolic screening to evaluate a whole host of newborn disease. So I'm going to try to walk through some of these. We have amino acidopathies, organic acidemias, and fatty acid oxidation and ketogenesis defects. Now, you might be a metabolomicist, in which case what I'm going to say might just hurt your ears a little bit, or you might be an everyday person, and these terms may not mean a whole lot to you. So I'm going to try to walk through those pieces a little better. Amino acids are, of course, the building blocks of proteins, but there are lots of biochemical pathways within humans that allow us to construct amino acids from others. So, and you probably will need the ability to break them down at some point to produce energy, for example. We sometimes burn protein for energy, right? So we see things like phenylketonuria, tyrosinemia, maple syrup urine disease, homocystinuria, hypermethanemia, and citrullinemia. I haven't tried to pronounce all of those out loud yet. So we see then that we have a variety of compounds associated with amino acid-like structures, and we need to be able to convert among them. And we can see that in some of them, like tyrosinemia, we have a problem associated with either the enzyme that leads to tyrosine or leads from tyrosine. So these are all amino acid associated conditions that can be found in newborns. The organic acidemias relate to organic acids. So we have glutaric aciduria, propionic acidemia, methylmalonic acidemia, and isovaleric acidemia. Okay, so some of these, again, are related to amino acids, things like glutaric aciduria. These are examples of other types of organic acids that are used as intermediates for constructing other compounds. So you might think to yourself, gosh, we have all of these effectively relate to a gene leading to or a gene leading away from one of these metabolites. Wouldn't it be simpler to just sequence these genes, right? But the truth of the matter is that screening for the presence abnormal concentrations of these particular compounds is a relatively easy thing to do with mass spectrometry. And you can see here I've included the label for Northwestern University here in South Africa. They have a great big human metabolome screening facility where they can screen newborns for this at a great clip. We don't have to send our samples off to Europe or to the States to have them processed. So all of this can be done at newbornscreening.co.za. That's a rather lovely facility to have here in our own country. And finally, fatty acid oxidation and ketogenesis defects. This point may seem a little confusing, but I would note that when you're my age and you have a little potbelly like this, you would like to think that something could possibly reduce the amount of these, the size of these fat cells in my body, right? And to do that, I'm going to need to do some exercise and so on, but my body is still going to need the mechanisms to break down fatty acids into things like acetyl-CoA and stuff like that that feed into the Krebs cycle to power all that energy I'm going to need for hiking up and down hills. We're going to do that, right? That's great. So we see then that if I have defects in the enzymes that allow me to do ketogenesis, that allow me to break down fatty acids, etc., I can do the exercise, but I'm going to not do anything to destroy the fatty acids I've been stockpiling this winter. I wanted to show one story here about phenylketonuria, which is a really interesting disorder in that if you are positive for phenylketonuria, there are certain substances you must avoid to eat. So in the United States, if you have a Diet Coke can, you'll frequently see on the side that there's this phenylketonurics contains phenylalanine. You see that? It's written right here in this tiny little print. So that tiny print is required by law to be on this because Coca-Cola, a Diet Coke in this case, has aspartame in it. And aspartame incorporates phenylalanine. If I, as a phenylketonuric, were to drink that, it would increase the concentrations of phenylalanine in my body, and I would have a reduced ability to get rid of it because my body has a defective enzyme for destroying it. So phenylketonuria is a big deal. How did we come to a place where we understood what phenylketonuria represented? Well, first off, you should know that one of the key outcomes of untreated phenylketonuria is abnormal mental development. So kids who just eat normally, who just eat things that contain phenylalanine willy-nilly, who have phenylketonuria tend to have pretty severe intellectual deficits that accumulate over time. So this is a terrible thing to have. If they knew, if I avoid these foods, I'm never going to develop this kind of developmental disorder, that's really important. So in 1934, we see that the mother of two children who had this intellectual impairment that developed over time went to visit a biochemist named Asbjorn Folling to figure out whether the strange smell of their urine were associated with their disease. Now, what kind of insight was that? One of the ways that our body's metabolites are reflected in us in phenotype is in our urine. We excrete metabolites all the time. So in this case, the fact that this woman's two children both had the intellectual impairment and had a strange musky smell to their urine flagged her to go talk to the biochemist. And that's how we learned what phenylketonuria is, how it results in these frustrating side effects. So this is an example of an inborn disease that we can detect through newborn screening. So if you're an adult, you've probably made it past all of these diseases. You know you don't have phenylketonuria, you don't have the impairments associated with these diseases. Why would you go to get tested for these? There's been a lot of research on this subject, so why would an adult choose to get genetic testing? The first is that I may know that people in my family characteristically get a particular disease more often than I hear of others. So in my family, we have a propensity to diabetes. So it might be that I have genetic markers that are associated with increased chance genetically for developing diabetes. Or it could just be something as innocuous as a set of alleles that mean that I really, really like candy. It could also affect. So here we see that I might know that my family has a history of this, and I want to be able to explain this in terms of the genes, the alleles that we are passing around. Next, I might like to guide my future medical management. It might even be that I'm going to change my diet. I'm going to stop polishing off a bag of jelly beans every week, and that's going to be good for me, right? So changes in diet and exercise can sometimes be motivated by people realizing that they have a genetic predilection for disease in response to these things. It may be that I want to allay concerns about my current family and potential children. Yes, if you're planning on starting a family with somebody and you come from a family that has a known history in, say, cystic fibrosis or other genetically related diseases, you might want to know, do you have a greater likelihood of producing a child who has genetic disease because of that? It's a very important bit of information. And finally, they might want to relieve uncertainty regarding diagnosis. And sometimes we think that the symptoms we're seeing are associated with a disease, but we're not really absolutely certain. So having genetic information can help us understand that better. We have genetic screening services, of course, in the United States and in Europe. But here in South Africa, we've started a genetic screening service of our own. So the Be Happy to Be You service, run out of CPGR in Cape Town, has begun genetic screening in a way that reflects the greater genetic diversity that we see in Africa. So this is a direct-to-consumer genetic screening. As I said, it's built out of Artisan Biomed here in Cape Town. They're measuring 700,000 SNPs. So single nucleotide variants, there are 700,000 of them that they can use to evaluate not the full sequences of every allele at every gene, but rather markers that are associated with risk in this case. So they're doing this not via sequencing technology. They're not just throwing all these samples into a next-gen sequencer. Instead, they're using an Illumina bead chip. This is kind of the successor to microarrays for measuring genetic variants. So in this, we're looking for markers that are known to associate with alleles, that are associated with disease risk, and they're very useful if you're trying to understand your ancestry. We have a lot of groups in South Africa that come from somewhere else, and understanding what fraction of your genetic markers come from where can actually be very informative for a lot of people. So and on top of this, it's helping to enrich what we know about African genetic diversity. Africa, in a lot of ways, has kind of lagged behind other continents in its adoption of screening for genetic variation. And with initiatives like this, we're getting to know a lot more about ourselves. Now, I wanted to mention Huntington disease. This is a marker that we first learned about in 1983. I've shown here the transcripts for the Huntington gene itself. And you can see that I've highlighted this region of yellow. These are CAG, 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 CAG repeats within the sequence. Ordinarily, for somebody who has a wild-type phenotype, we see fewer than 36 copies of this CAG sequence within the transcript. That's grand. However, we note that when people have more than 36 repeats, it increases the likelihood that they're going to develop Huntington disease. So, in some cases, we see that people at a relatively young age, 35 to 45, are first being diagnosed with this disease, and after around 15 years, the disease is most frequently fatal. So this is a marker that you would like to know about then. If you're developing the symptoms of Huntington disease and you suddenly discover that you've got 100 copies of this repeat in that gene, the severity and the progression of this disease may be equally, commensurately more difficult. We also see that different cells in your body are going to have differences in this gene. That if you look at the Huntington gene in your skin cell or in your liver or in your brain or in your kidney, that you see different numbers of repeats depending on where you look, even though it's all your genome. So here we see that this gene can actually kind of slip when it makes copies of itself to create more CAGs that fall within the transcript. That leads to this variation. Now I want to bring with it a key ethical concern when we think about this in the context of genetic counseling. If we were able to tell you how many repeats you have in your Huntington gene, that would be informative to you about whether or not you are likely to get sick with this. But today, as of now, we do not have a treatment for this Huntington disease. And that way you might know that you're dying, but we wouldn't have anything to tell you that could really help you cope with that disease or forestall your death. So learning that you have this particular marker is not a particularly useful bit of information, except in doing things like planning your estate. That's a harsh reality. Most of the genetic traits we're talking about have something that we can do to mitigate the circumstances, but it's not true for all. So I thought that this was a really valuable insight that came along with it. I'm just going to read this off the slide. This was from a study on who benefits from, who takes part in genetic counseling for adults. Our group, similar to other studies, showed a general predominance of females requesting testing. And we had more females present for testing than would be expected based on the general population. Multiple potential reasons for this predominance have been proposed, including that women are more invested in the reproductive decision-making process and the rearing of children. Women are more willing to make difficult decisions and deal with the consequences of those choices, and women may be better able to cope with negative results. I just think men need to step it up a little bit. Women are the ones who most frequently ask for genetic testing and who figure out how to adjust once they receive that information back. Men can take responsibility in their families too. Maybe we should think about that. All right. Does this have real consequences for people? Do people make big life-changing decisions based on this kind of testing? They assuredly do. The BRCA1 gene was, we learned about a particular allele for it in 1994 by somebody who was on one of my graduate school committees, Mary Claire King, actually. Lovely person. One of the things that she discovered was that people who have this allele at the BRCA1 gene are much more likely to develop familial breast cancer. So this is not just somebody who has no family history. This is somebody where many women in the family are known to have died from breast cancer. These are the folks who are really in this hot spot for getting tested for what allele they have at BRCA1. So it's a predisposition to breast cancer and other cancers. It's not just the one. And we see that there are two different genes, BRCA1 and 2, that have this strong association. So I think many people know the actress Angelina Jolie. She did the screening because she knew that a lot of people in her family had died of breast cancer, and learned that she was in this very unhappy demographic, the group of people who have two disease alleles at BRCA1 or 2. I forget which one. So she wrote a really fascinating article that appeared in the New York Times to talk about her decision in response to this. And she decided to have prophylactic mastectomy, which is to say that she wasn't sick, but in order to protect herself in the future, she decided to have this breast removal surgery in order to forego the increased chance of breast cancer. She said in her New York Times article, "My chances of developing breast cancer have dropped from 87% to under 5%. It's clear then that people make pretty wide-sweeping decisions in response to the genetic information that they get. I want to talk about one more disease before we stop for intermission. This is celiac disease. So celiac disease is essentially an autoimmune disease that people when they get exposed to say bread that's made from wheat or rye or barley, respond really badly to the presence of the gluten protein present in this, and that the appearance of gluten causes their immune system to launch an immune attack on that gluten. But the immune attack has another barb. In attacking the gluten protein, it also attacks the lining of the intestine. And it's much more than, oh, my belly is tummy. It sends people to the hospital and can kill people, in fact. So celiac disease is a very, very big deal. But what do we know about its genetic relationship? For that, I would highlight two different haplotypes known to be associated with this, DQ2 and DQ8. These make changes to what's called an antigen presentation complex, we're seeing a picture of it over here. So we have two different proteins that come together to make a complex, and they form this groove in which a gluten peptide sits. In effect, antigen presentation is a way for the immune system to train itself. When an antigen is presented, antibodies that your body knows how to make can be created and amplified. So in this case, if gluten is being presented preferentially by this person's immune system, they're going to generate lots and lots and lots of antibodies that know how to recognize gluten. So DQ2 and DQ8 haplotypes are much more likely to grab onto gluten and show it preferentially as, this is a threat, take this seriously. As a result, these folks have immune systems that are then trained to attack not just gluten, but the person's intestines themselves. And with that, we come to intermission. In just a second, we'll start up again with acquired mutations. And welcome back. So in the first part of this talk, we spoke about the inborn genome as a predictor for potential disease. In the second part, however, we're going to spend our time talking about acquired or sporadic mutations. So this is about the change in our genomes over time. That might seem like a very foreign concept, so we're going to spend a little time on three different big examples. We're going to start by talking about cancer and the tumors that form from it, and the acquisition of mutations. In the second part, we'll talk about our other genome, about the mitochondrial DNA, and the concept of heteroplasmy and its effect on neurodegenerative disease. In the final case, we're going to talk just a little bit about infectious disease as well. So up here I've shown a pathology slide from University of Pennsylvania. These are showing the new vasculature, the new blood vessels that are forming as a cancer progresses within a tissue. In the second case, I'm showing a schematic diagram of the mtDNA, the mitochondrial DNA, that we find in all of our mitochondria. And at the bottom, I'm showing a molecule called neuraminidase, which is probably better known by its initial, by N, when we talk about different influenza variants. The N1, H1N1, for example, is neuraminidase 1. So I'm going to start by talking about cancer as a disease of mutations. When people have cancer, they frequently lose, in at least in just the tumor, the ability to control the formation or the accumulation of new mutations within the genome. So cancer is a disease that is driven by particular mutations in critical systems, and from that it gains a whole bunch of extra mutations. So we speak of these as driver mutations and sometimes passenger mutations. I've chosen this angry driver to show that these are driver mutations that no one would really want. The researchers in this 2018 article put together a lot of data that existed out there that had been produced in part by groups like TCGA, the Cancer Genome Atlas, to analyze which genes are most frequently playing a role in the formation of a cancer or of a cancer becoming more aggressive. And these were the 5 genes that, for which mutations within them were most likely to drive a new cancer ahead. So I think TP53 is a very widely known gene at this point. It is a tumor suppressor. So when it detects stresses, it is able to regulate the cell cycle, prevent, for example, cell division if it knows that the genome is a little out of order. And so as a tumor suppressor, its job is to prevent the cell from dividing without end when certain things are not going correctly. So it's kind of a checkpoint. So TP53 is one of those genes that if a cancer is able to distort the action of TP53, it may make driving forward to become an aggressive tumor much easier. Okay, PIK3 kinase is a kinase. And kinase is a term that is very familiar in molecular biology. A kinase is a protein that adds phosphorylation to other proteins. Very frequently, this is part of a cell communication process to launch off a cascade of some sort to dramatically change cell behavior. So the PIK3CA is an oncogene, which is to say it's a gene that participates in cancers. It is a kinase. It's associated with the AKT and mTOR pathways. If you're a cancer biologist, those two pathways are very, very common terms that will crop up. KRAS is an oncogene, and a GTPase. GTP is can be thought of as kind of a power source for driving molecular reactions. So it's able to function in cancer by using up GTPase, and it has isoforms that misbehave in the wrong tissues. So it is a gene for which multiple isoforms are possible, multiple ways to stitch together its exons. And in tissues where you wouldn't expect to see it, in cancer, this gene can suddenly start being expressed in places it never was before in some of these weird isoforms, and it really messes with the cell's activities. P10 and ARID1A, I'm not really going to go through all of these, but I would point out that all of these typically have some sort of interaction with cell cycle and DNA. So in cases like this, we see that we're talking about a helicase and chromatin remodeler. If you are unwinding DNA or winding DNA, or you are remodeling the chromatin, making the DNA more tightly packed or less tightly packed, you're altering how genes are expressed within that. So you can see that there are lots of ways that these very active proteins could play a dangerous role. You know, in normal sequences with normal regulation, these genes are quite essential to the daily work of a cell. But if you develop mutations within them, the way that things can go badly can lead to very aggressive cancers. So these genes are not ones that we can tolerate mutations in very well at all. So one of the things that has really driven precision medicine to be associated with sequence everything is cancer tumors. That when we have tumors, understanding which sequence variants they contain helps us to understand how aggressive the disease may be, and they may give us some hint as to which clinical decisions we need to make. So I would start with breast cancer. We've already talked about BRCA1 and 2 as genetic locations where the alleles have a real impact on whether cancer develops or not. But when we look at breast cancers, one of the most common reasons that we are going to seek out more sequence information is to understand a trio of receptors that we care about. The estrogen receptor is abbreviated ER. The epidermal growth factor receptor is sometimes called HER. And there's another one here, the progesterone receptor. So when we say that a cell is ER positive, that is to say that it does contain the estrogen receptor. When we say that a particular tumor is HER2 negative, we are saying that it lacks the receptor for epidermal growth factor. So these are useful to know because one of the most common kinds of treatment for breast cancer are hormonal treatments. And if you're using a hormone for which this tumor lacks a receptor as a way to tell those cells to calm the heck down, no one's listening, right? So we don't want to waste people's time and money and the awfulness of therapy with these things if the tumor isn't going to listen to those signals anyway. So if you just consider the HER and the ER, you can see that both could be positive, wild type essentially. One could be positive and the other negative. One could be positive with the other negative, or they could both be negative. And that's what gives us these four types of cancer, of breast cancers, called luminal A, luminal B, HER2 overexpression, and basal-like. So HER2 overexpression might superficially look relatively normal. It's got a low estrogen receptor here, but there's an overabundance of the expression at epidermal growth factor receptor. One of the tricks that these little receptors can do is that they can become constitutively active. So if EGFR is is modified to always act as though epidermal growth factor has arrived at the cell. It's constitutively on, and so it's continually signaling the cell, grow, 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 grow. That is another way of saying that you have uncontrolled cell growth is cancer. The two terms are very closely related to each other. So I would note that this basal-like type of cancer, of breast cancer, sometimes appears in a form called triple negative, which is to say that not only is ER negative and HER2 is negative, but progesterone receptor may also be negative. And in a case like that, we have a phenotype of breast cancer that's known for particularly aggressive growth. So if you lack a receptor, using a hormone to treat the tumor isn't going to do a lot. So as we see that these genes are negative in the cell lines, our ability to use hormone treatment recedes. There are other treatments available, but this is but understanding what subtype of cancer we're dealing with in a breast tumor has dramatic differences for how it would be treated. Now, I spent a lot more of my time at Vanderbilt at colorectal cancer types instead. So one of the types that we look for is called a hypermutator, a type of colorectal cancer that is very, very rapidly accumulating mutations. This may be because of microsatellite instability, which means that it's able to develop repeats a lot more than it was in the past. It might be due to hypermethylation, which can, of course, alter the gene regulation, the expression that we see in its genes. Or it could be that we see deficits in MLH1, the silencing of that gene. So this is part of the DNA mismatch repair, and if you're not repairing mismatches in your DNA, you are accumulating mutations at a much higher rate. Now, there's kind of a paradoxical effect that we see in hypermutated cancers. We see that they're not a huge fraction of all colorectal cancers, but when you see a hypermutated phenotype, you see that this tumor is developing neoantigens. It's developing variant versions of its own proteins that may be recognized by the immune system as not self anymore. That the tumor, in effect, begins acting like a foreign body within you that your own immune system can then attack. So in some cases, this is actually an opportunity for immunotherapy. There is a world of information out there to understand about genes and the variants, the variant alleles that are present for them that may have an impact on how the disease should be treated or on the phenotype of a particular cancer. Now, in my last couple of years at Vanderbilt, I crossed over with some of the researchers who were coming in to develop My Cancer Genome. It's a really cool resource. It's available to anybody. You don't have to be part of Vanderbilt to use it. But I really love this description of all the things that they're offering to try to make precision medicine or patient-centered medicine more feasible in the context of cancer. My Cancer Genome is a precision cancer medicine knowledge resource. It's intended for physicians, but also for patients, for caregivers, and for researchers, which is, I think, mostly us. My Cancer Genome gives up-to-date information on what mutations make cancers grow, drivers mutations, and related therapeutic implications, including available clinical trials. So if you develop this disease and you want to be part of a research trial, this is one of the ways that you can do it. My Cancer Genome is a one-stop tool that matches tumor mutations to therapies, making information accessible and convenient for busy clinicians. Because after all, at least in the United States, your doctor doesn't have time for just you. Your doctor is sharing their brain among 50 patients at the same time and trying to understand what the best course of action for your tumor could be is something that can be helped if you've got the right information to help you. Okay, so clearly we see then that what sequences acquire mutations in the context of cancer is hugely important to understanding the likely progression of the disease and strategies for how we would treat it. Mitochondrial DNA is also quite important for our developing disease, and it's something that a lot of us don't think about. Our copy of an individual cell contains one nuclear genome, but on top of that, it might have 1,000 mitochondria in it, that one cell. And if each of those thousands of mitochondria has its own genome. Now, it's a genome that doesn't look like ours. It's not a whole bunch of chromosomes in little pairs and so on. Instead, it's a genome that looks very much like a bacterial one, a single loop, basically. And it's only 16.6 kilobases long, so it's quite small, quite compact. The genome of mitochondrial DNA is quite essential because it codes for products necessary in oxidative phosphorylation. This is the process by which we generate ATP. So if you can generate enough ATP, you can power the cell. If you fail to generate enough ATP, then some cell processes may go starving and not be able to go forward normally. Okay, so oxidative phosphorylation is notable for its production of reactive oxygen species. I think we all hear about the foods we should eat must contain antioxidants to avoid reactive oxygen species. So I think that's a familiar concept for people. But there's a lot of oxidation and of oxphos taking place within cells all the time, and this is just one of the necessary offshoots of that process. So if your cell contains 1,000 mitochondria, you might think you have 1,000 copies of exactly the same empty DNA molecule. This is not, however, true, because frequently we find that to begin with, we have cells that, as infants, we have cells that have all pretty much the same mitochondrial DNA. We find that over time, subsets of our cells have an increased fouling of the mitochondrial genome. And a lot of people think that there's a strong link between the effects we see in aging and an accumulation of dysfunctional mitochondrial genomes. So in a given cell, if you have a bunch of mitochondria, some of which are perfectly functional and others of which have flawed mitochondrial DNA, you have a condition called heteroplasmy. Now, I've already mentioned the term mosaic before, which suggests that different cells have different genotypes in effect. In this case, you may find that cells in one part of your brain have a higher proportion of flawed mtDNAs, whereas in your skin cells you have a very ordinary collection of perfectly working mitochondrial DNA. In a case like that, if you're looking within an individual cell, you're talking about heteroplasmy. When you talk about different cells having different levels of functional mitochondria, you're really talking about a mosaic, where you as one organism have different sets of mitochondrial DNAs depending on where in your body you're speaking. Okay, so there's a bit of controversy tied to this, but I think much of what I'm saying on this slide is very well borne out. I think that when I was in graduate school, there was a lot of belief that flaws accumulating in mitochondrial DNA led to aging, which is why we feel tired and eventually die. I think that simplistic view of aging and mitochondria is now pretty passé, and most people would say that's very much overblown. But there is good reason to think that some of the chance events that happen over time in our mitochondria do have links with real disease, and neurodegeneration is probably the number one way in which these mitochondrial DNAs have bad effects on us. So, why is it that we think that mitochondrial DNA mutations lead to neurodegenerative disease? Over the years, the replication that we carry out, because mitochondria do division and create two copies of a mitochondrion from one, so this process of replication can introduce somatic mutations that in our body's cells, we find that errors accrue in mitochondrial genomes. There's a very common set of deletions that we see, particularly in the aged and particularly in the brains, that can reduce the ability of the mitochondrion to carry out its gene expression normally. So brain regions such as the substantia nigra are associated with mitochondrial DNA damage in aging. So this is not true for everybody, but for some folks, we find that their ability to generate the fuel needed for the cell to run is diminished particularly in certain brain regions. And this substantia nigra is a rather important area. So there are three major types of disease that have been associated with mitochondrial DNA damage in the brain. So Lewy body disease might be kind of an unfamiliar term, but I think many people have heard of Parkinson's disease. So Parkinson's disease is something where you get rigidity and your muscles don't respond quite as well. You may have central tremor, etc. It's a very uncomfortable disease to develop for somebody. So we believe that diseases like Parkinson's take place because of the accumulation of DNA damage in mitochondria in particular parts of the brain. So that's one danger. Alzheimer's disease, I mean, we talk a lot about how a prion is associated with this in the formation of plaques in Alzheimer's patients, but it's also the case that we've observed some mitochondrial DNA damage that may be connected with the production of Alzheimer's disease. Of these three, I think stroke is the one that I'm least sure about. There are some papers who argue definitely for it and some papers that are a little less sure about this, but it appears that mitochondrial damage to brain cells may result in an increased chance of stroke. That is obviously a rather dramatic event to go through. So these are three major neurodegenerative diseases, all of which may be associated with the accumulation of mitochondrial DNA variations. Now, I ask you, how effective is this as a biomarker? Are you able to sequence mitochondrial DNA from cells from deep inside the brain? Not really. So this is again a case where simply sequencing your way to an answer is not going to be that effective. To know that there are mutations within within the mitochondria of a part of the brain might be very diagnostically informative, but getting that information becomes quite difficult. So it might be that instead we need to think about ways that we could evaluate what would be the effects of mitochondrial DNA deficits. Would we see changes in energy production in the brain that could be visualized? Can we use imaging technologies to learn about the rate of turnover or the rate at which ATP is being generated in cells? Simply sequencing our way to an answer doesn't really work when you're dealing with cells that are buried in the middle of your brain. I think many of us fail to use one of the greatest public health inventions of the last centuries. Vaccines have saved more lives than we can easily think about. And the flu vaccine is one that I so frequently hear poo-pooed that I'm bothered that we're not taking advantage of it. The flu is a really dangerous disease, and I think that this year of COVID-19 has really demonstrated to us that respiratory infections can be much worse than just a cold. The influenza carries off a huge number of people every year, and we can protect ourselves against that by taking the vaccine on a yearly basis. It may be that the life you're saving is not your own, but your grandparents'. But we should all think much more seriously about taking the flu vaccine that's available to us. But why do we have to take a different shot for this every year? You were vaccinated for measles when you were a child, but you don't have to get vaccinated for it now. And the reason why is that influenza is a disease that can change its molecular appearance very, very rapidly. So we start with the fact that animal reservoirs can be the source of new strains of influenza, that birds and pigs in particular may be infected with a virus that crosses over, because it's a zoonotic disease, that can cross over from animal populations into human ones, which means that although the virus may be evolving separately in human and animal populations at a given time, these crossover events lead to dramatically different versions of the virus making an appearance in human populations when they've only been seen in pigs for a long time, or in the case of COVID-19, in bats. So, that's a big problem. The other one is that the virus is able to acquire new mutations just in the process of passing around in human populations. We sometimes speak of this as antigenic drift, that you might have developed antibodies against last year's flu when you had it. And so when you are infected with this year's flu, if it were exactly the same sequence, your immune system would immediately defeat it, and you'd have the virus wouldn't have a chance to spam the world with 1,000 copies of itself. So that's one reason why we have a strong selection pressure. You remember your terms from evolution, I think, that we have a selection pressure that if that virus doesn't change its sequence, it's much less effective in infecting a population in a new season. So there are lots of reasons why we see that influenza is able to make a lot of changes in its sequence from year to year. At present, there are two different vaccine pools that are in motion at any given point in time. That's because the flu season in the southern hemisphere is 6 months out of phase with the flu season in the northern hemisphere. If you live in the tropics, well, the flu is always kind of around, so that's not so great. So in this case, we have two different processes going, where a flu vaccine is being prepared for the northern hemisphere, 6 months out of phase, with the same process going on in the southern hemisphere. And the WHO has a very active working group that's working to decide which strains at the end of each flu season are most likely to make a reappearance in the coming year. At present, the flu vaccine contains often 3 different strains to help protect us against the variability that we might see in oncoming years. So we see that 2 different types of influenza A, H3N2, you remember I mentioned the neuraminidase is the N. The other part is hemagglutinin. Hemagglutinin is the protein that allows the virus to sort of attach itself to cells. This is a little image of it down here. So the influenza A strain that contains version 3 of the hemagglutinin and version 2 of the neuraminidase is one that gets included in the vaccine. And the other one is H1N1, a hemagglutinin of type 1 and a neuraminidase of type 1 that gets put into this vaccine. But influenza B is also in circulation. So we have these major strains and minor variants within those strains that we all have to protect against. Having 3 different viruses attenuated in these vaccines helps us to mount a defense against all of them if we are to encounter them. Okay, so what we see then is that 6 months before the flu season, the WHO is deciding which sequences are going to represent these A and B strains in the batch that people get next year, the batch of vaccine. And I think that's an amazing body of work, to be able to kick off this industrial process by which a huge number of vaccines will be available in time for the next year's flu season that represent the sequences seen in the prior one. Now, to speak of this just in the context of viruses is to miss an important point. Here in South Africa, at least, tuberculosis is a very, very big deal. Ordinarily, we can treat somebody for tuberculosis for about 6 months and it's simply gone. Things do not always go this way, though. There are lots of reasons why people may be infected with strains of TB that are resistant to those drugs, or we might find that their adherence to the drug schedule is so imperfect that they allow the tuberculosis to rally back by mutating and gaining an indifference to these drugs. So a sensitive TB can become a multiply drug-resistant or extensively drug-resistant form by acquiring mutations. That we see that the genome is acquiring mutations that allow it to escape from the selection pressure it's under from the antibiotics of treatment. Now this diagram comes from this 2014 paper. It represents a person who had immigrated from Eastern Europe into Norway, and they had been detected to have TB, went through treatment, and instead of their treatment lasting 6 months, it lasted 45 months. 45 months, 4 years of treatment for this disease. We see that at first they were treated with a combination of isoniazid and rifampicin. That's a very common kind of treatment. But after 8 months, they were still having a lot of problems with the disease, and it was detected by sequencing the bugs that were growing in their lungs that this bug had developed fixed mutations, meaning more than 85% of the bacteria growing in this person's lungs were now resistant to INH and resistant to rifampicin because they'd acquired mutations against it. And they were fixed in that population. No amount of treatment with rifampicin or isoniazid was going to do anything to kill this thing, to kill these bugs. So they started switching to different treatments. We see that after 14 months, they had developed a mutation in the RRS gene, which caused them to become resistant to streptomycin, which is one of the fallback treatments. So we can see that all of these different drugs ended up getting used in the case of this one patient. I've got 11 different drug names here, and there are more just in this little test set that they were using. This chase that MTB led the doctors over the space of 4 years resulted finally, not in death, but in the person becoming culture negative, effectively being cured, that they were no longer able to culture TB out of that person's lungs at the time of 45 months. However, the damage that had been done in that process meant that 3 months later, they had to get a pulmectomy to remove so much of the damaged tissue from this person's lungs. Certainly, microbial disease can become resistant to the drugs we use to defeat it because of its ability to create variant forms of these genes that are no longer susceptible to the drugs. So MDR and XDR tuberculosis are a hugely painful part of dealing with the tuberculosis pandemic that the developing world will be for years. All right, lots of takeaway messages from this lecture, but I've chosen some pretty simple ones. Generally, the alleles you carry do not determine your fate. It might be that they predispose you to particular diseases, but generally speaking, they're not all in the case of Huntington's disease. Generally speaking, there are things that we can do to mitigate our chances of developing these diseases. You may have been born with a DNA variant, but you might acquire it by a variety of means. Certainly when we see cancers and all the mutations they acquire, that's not a normally functioning cell anymore. Its ability to accumulate mutations is far greater than the ones that appear in most of our bodies. Okay, so it's also the case, however, that you don't have to be a human cell to acquire new mutations. You may be a virus, or you might be a bacterium, and they can certainly gain new mutations, which causes them to become resistant to the therapies we would direct against them. When we say that every cell in the human body contains exactly the same genome, well, we're telling a little lie, aren't we? We saw several occasions in which mosaicism may take place, or heteroplasmy may take place. If you develop a tumor in a part of your body, that tumor may vary quite a lot from the rest of your genome. So this degree of genetic variability is something that we must take into account when we're understanding the causes of disease within human populations. Hopefully, this has helped you to understand a little better about how these genetic changes can function as clinical biomarkers.